Rabies is of the genus Lyssavirus and of the family Rabbidoviridae, which includes other related viruses that infect mammals and arthropods. It is a neurotropic RNA virus, which means it infects nerve cells. The structure of the RNA is single-stranded, negative sense, unsegmented, and enveloped. As previously stated, rabies is negative sense. Positive sense viral RNA is similar to mRNA and can be immediately translated by the host cell, whereas negative sense viral RNA is complementary to the mRNA, and therefore it must be converted back to positive sense by RNA-dependent RNA polymerase before translation. Rabies typically affects poor and vulnerable populations. Affluent areas have access to vaccines, which prevents the potential hosts from contracting the virus in the first place. In humans, rabies typically presents in 5 stages: incubation, prodrome, acute neurological period, coma, and then death. Incubation has been observed to be around 1 to 3 months clinically. This can be highly variable from person to person, ranging from as little as 10 days to 1 to 3 years. Clinically, children have shown to have a shorter incubation time period. If the bite that introduces the virus to the body occurs closer to the central nervous system, then the incubation period might be expedited. Incubation is the time from exposure to the onset of signs and symptoms. Next, prodrome stage typically lasts around 2 to 10 days. Clinical symptoms typically begin during this stage. However, these symptoms are very nonspecific. They range from malaise, fever, fatigue, sore throat, cough, difficulty breathing, nausea, vomiting, and headache, to agitation, photophobia, priapism, increased libido, insomnia, nightmares, and depression. This range of symptoms can typically be confused with encephalitis. A distinctive symptom is pain and burning sensation at the site of the bite. Direct fluorescent antibody test can be used for the diagnosis of rabies. It allows for the detection of Rab-V antigens via fluorescent microscopy. With the correct conjugate, this test is nearly 100% accurate. Unfortunately, fluorescent microscopes are very expensive, and the poor and vulnerable populations that are disproportionately affected by rabies are not able to use them. More research has been conducted over the past few years and is continuing to be conducted currently in order to find a cheaper alternative. Other options, such as the direct rapid immunohistochemistry test and the indirect rapid immunohistochemical test, have been suggested as surrogates for diagnosis. The acute neurological period is when idiosyncratic symptoms truly start appearing. Fever, burning sensations, rigid neck, muscle twitching, focal and generalized convulsions, rapid breathing, and hypersalivation may occur. If the predominating symptom is hyperactivity or hydrophobia, then the patient's case is classified as furious, whereas if paralysis is the predominating symptom, then the patient is classified as paralytic. Towards the end of this phase, rapid and irregular breathing begins, and paralysis and coma follow. In the 4th phase, coma, respiratory arrest may ensue without the help of ventilatory assistance. With assisted breathing, the patient's prognosis may be extended by days, weeks, or even longer. However, the vast majority of rabies cases usually end in death, the 5th phase. Usually death due to complications occurs within 3 to 7 days after onset of symptoms. Rabies has a near 100% fatality rate, causing around 59,000 deaths per year. There have been at least 7 reported cases of recovery documented. And while not all cases can end in recovery, life support measures are always used for every patient, and all options are exhausted in trying for survival.
Rabies is typically found in the saliva of a rabid animal, which, when this bites a person, travels up the peripheral nerves and to the spinal cord and the brain. The person infected with rabies is then unable to swallow and typically experiences a choking sensation when they try and drink. This is due to the muscle's inability to control swallowing. Consuming little water can concentrate the virus in the mammal saliva, which also can be beneficial for the virus so it's not flushed out. The path of infection for rabies typically starts out with a person who is bitten by a rabid animal. The virus enters through the infected saliva, which enters the wound, and then the rabies virus travels through the nerves to the spinal cord and brain, through endocytosis through the muscle cells, which is followed by the neurons. When it reaches the brain, the virus multiplies rapidly and passes to the salivary glands. This is when the person begins to show signs of the disease. The infected animal then usually dies 7 days or so after becoming sick. From research on the rabies vaccine, the protective antigen in the rabies virus was found to be a glycoprotein. This glycoprotein was located on the spike surrounding the virus. Rabies was then treated with tri-N-butyl phosphate, which broke the virus into many pieces and disrupted the lipids within the virus. So through the process of vaccinating, rabies has been developed in people due to environmental and evolutionary errors that could not be controlled. So there are two main types of rabies vaccines that are used today. The first one is called the human diploid cell vaccine. So this vaccine works by exposing you to a small dose of the virus, which then causes the body to develop immunity to the disease. In this vaccine, the virus is taken from the MRC5 cells in which the virus was grown. The second vaccine used is the purified chick embryo cells. And so both of these increase the amount of neutralizing antibodies which target the glycoprotein found on the surface of the virus. So upon injection, the beta-propiolactone is utilized in order to inactivate the virus. The response of the antibodies is then developed against the glycoprotein found on the surface. These vaccines are indicated for post and pre-exposure prophylaxis to prevent human rabies. The rabies immune globulin is used with the rabies vaccine in order to prevent infection that is caused by the virus. So this works by giving the body antibodies needed to protect and slow the progression of the virus until an immune response is capable of being developed. And this process is known as passive protection. And so the human rabies immune globulin is safer for human use because the vaccine is needed in higher doses for a desired response, which can lead to hypersensitivity in our nervous system. And the rabies immune globulin is taken from donated human blood serums. And although these blood donors are screened for diseases such as hepatitis and HIV, there still can be a chance that they can transmit these bloodborne infections.
The viral genome of rabies encodes for 5 proteins: the nucleoprotein or the N protein, the phosphoprotein, also called the P protein, the matrix or M protein, the glycoprotein or the G protein, and the polymerase protein, also called the L protein. Rabies are bullet-shaped and enveloped with G protein lipid bilayer and 300 to 400 spike proteins, each made of a G protein trimer, which many are glycosylated, and these trimers are tightly arranged on the surface of an envelope. Underneath these spikes is the matrix or M protein on the antivirus envelope, which is tightly folded and is the link between the lipid bilayer of the virus envelope and the ribonucleoprotein core. The ribonucleoprotein core is made primarily of the RNA genome bound by the virus nucleoprotein or N protein to form the nucleocapsid core, and along this in the RNP or the ribonucleoprotein core, there is also the polymerase protein, also called the L protein, and the phosphorylated protein, called the P protein, which will later make the transcriptase complex, which is involved in all polymerase activity during the genome replication. It is important that the RNA virus, especially since it's a negative strand, it is important that it has an RNA-dependent RNA polymerase in its virion, so it ensures that all the negative strand RNA becomes infectious, because a negative strand on its own cannot be translated by the ribosomes into proteins. So the RNA-dependent polymerase will help translate it into a positive strand, which then can be made into the viral proteins to replicate and create more virus, which then can be budded off to other parts of the body. When the virus comes upon a nerve cell, where it is most commonly to bind, especially in the brain, but it can also bind to muscle cells, but when it comes on to the nerve cell, it is most commonly going to attach to 3 receptors: the nicotinic acetylcholine receptor, the neuronal cell adhesion molecule, and the P75 neurotrophin receptor. The nicotinic acetylcholine receptor is an ionophore, usually functioning in muscle contraction by allowing cations to flow through when triggered by acetylcholine. The neuronal cell adhesion molecule is important in neural development and is usually bound by heparan sulfate. And the P75 neurotrophin receptor is important in controlled cell death, also called apoptosis, and survival and migration and axonal growth inhibition, and is usually triggered by toxic peptides like amyloid peptides or neurotrophins. The part of the virus that binds to these receptors is the G protein spikes. They are the most effective to binding to these receptors, as I said, also specifically in nerve cells. And then once it binds, the virus will be taken in via endocytosis into a cell called the endosome. Then the rabies envelope will fuse with the host cell membrane and it will penetrate the host cell through pinocytosis. The virus cell will then release its RNP into the cytoplasm. The nucleoprotein will dissociate from the ribonucleoprotein capsid and expose the ribonucleoprotein core, which is important in viral RNA synthesis. First, the viral single negative-stranded RNA genome has to be transcribed into messenger RNA for replication. A viral-encoded polymerase, the L protein, transcribes the genomic strand of rabies RNA into a leader RNA and 5 capped and polyadenylated mRNAs, which are the 5 previously mentioned proteins. They have the methyl caps and poly-A tails in order to prevent them from being degraded while they're in the cytoplasm. This mRNA is then translated into 5 proteins, which were previously mentioned, are going through the endoplasmic reticulum, and they are glycosylated, and the synthesis ends in the Golgi apparatus. The intracellular ratio of the leader RNA to the nucleoproteins, the N protein production, regulates the switch from transcription to replication. So when the switch is turned on, replication of the viral genome begins once enough transcription has occurred. The first step is synthesis of full-length copies of the viral genome, but these are anti-genome copies, so they're going to be positive strands. So when the virus wants to replicate, it first has to create a positive strand, and this will become non-stop. All the stop codons will be ignored, like the stop codons between the proteins that coded for each of the proteins. It will just create a straight through positive strand of the RNA virus. Then the viral polymerase will enter a site at the 3' end of the genome and proceed to synthesize the copies of the genome. These positive strands of rabies RNA serve as templates for synthesis of the negative strands of the viral genome to replicate it so that it can have more of itself. Once more negative strands have been created during the assembly process, the NPL complex encapsulates the negative stranded genomic RNA to form the ribonucleoprotein core, and the M protein forms a capsule or a matrix around the RNP core. The RNP-M complex will migrate to an area of the plasma membrane of the host cell plasma membrane containing glycoprotein inserts, and the M protein will initiate coiling. Then this RNP complex will bind to the glycoprotein, and the completed virus will bud from the plasma membrane within the central nervous system, which is why this causes the disease. Therefore, the process of infection of a rabies virus into a host cell membrane can be summed up into 9 steps.